This is part 2 of Chapter 6, the Skeletal System, Bones and Bone Tissue. In this video, we will cover bone formation, including intramembranous and endochondral ossification. We will also briefly touch on bone growth. Ossification or osteogenesis is the process of bone formation. It occurs in 4 situations: during embryological and fetal development, when bones grow before adulthood, when bones remodel, and when fractures heal. Bone formation occurs in 2 different ways. Intramembranous ossification occurs when the embryonic connective tissue membrane, which is the mesenchymal cells, are replaced by bone. Endochondral ossification occurs when the embryonic hyaline cartilage models are replaced by bone. Let's begin with intramembranous ossification. This is the way that flat, short, and irregular bones are made. As a reminder, these types of bones include some of the skull bones, part of the mandible, and the clavicles. Intramembranous ossification occurs in embryonic mesenchyme. Centers of ossification are locations where the membrane, or in the membrane where ossification begins. These centers expand outwards to form bone by ossifying the membrane. Fontanelles are what are known as soft spots on infants' heads. These are membrane-covered spaces between the developing skull bones that have not yet ossified. Ossification of fontanelles are typically completed by the age of 2. There are 3 steps to intramembranous ossification. The first step is osteoblast formation. Some embryonic mesenchymal cells in the connective tissue membrane differentiate into osteochondral progenitor cells, which then form osteoblasts. The osteoblasts produce bone matrix, which surround the collagen fibers of the connective tissue membrane. Once embedded in the bone matrix, the osteoblasts become osteocytes. As a result of this, many tiny trabeculae are formed. The second step in intramembranous ossification is the formation of spongy bone. In this step, additional osteoblasts gather on the surface of the trabeculae, where they produce more bone matrix. This results in the trabeculae becoming larger and longer. Spongy bone is formed when the trabeculae connect and form a scaffolding network separated by spaces. The third and final step in intramembranous ossification is the formation of compact bone. Cells in the spaces of spongy bone specialize to form red bone marrow. Also, the periosteum is formed by cells surrounding the developing bone. Lastly, osteoblasts in the periosteum lay down bone matrix that forms compact bone. The end result of intramembranous ossification is a sandwich of spongy bone between 2 layers of compact bone. This is what we see in flat, short, and irregular bones. The second manner in which bone is made is through endochondral ossification. If you have taken medical terminology, you learned that the root chondr means cartilage. So this way of making bone relies on a hyaline cartilage model. Endochondral ossification is used to make the bones of the base of the skull, part of the mandible, the epiphyses of the clavicles, and most of the remaining bones of the skeletal system. Endochondral ossification occurs in 5 steps. The first step is the formation of the cartilage model. This occurs around embryonic week 9. Embryonic mesenchymal cells aggregate in regions of future bone formation. These mesenchymal cells differentiate into osteochondral progenitor cells that then become chondroblasts that produce a hyaline cartilage model with the approximate shape of a future bone. As chondroblasts are surrounded by the cartilage matrix, they become chondrocytes. The cartilage model is surrounded by perichondrium, except where a joint will form with another bone. The second step of endochondral ossification is the formation of a bone collar. This occurs through embryonic month 3. Blood vessels invade the perichondrium surrounding the cartilage model, causing endochondral progenitor cells in the perichondrium to become osteoblasts. Osteoblasts create compact bone on the cartilage model surface, forming a bone collar. The cartilage model increases in size due to interstitial and appositional cartilage growth. The chondrocytes in the center of the cartilage model absorb some of the cartilage, enlarge and release matrix vessels, which starts the formation of hydroxyapatite crystals in the cartilage matrix. At this point, the cartilage is calcified. The chondrocytes die, leaving enlarged lacunae in the calcified matrix. The third step of endochondral ossification is the formation of the primary ossification center. This occurs during embryonic month 3 through birth and adolescence. Blood vessels grow into the enlarged lacunae of the calcified cartilage. Osteoblasts and osteoclasts move into the calcified cartilage area from the periosteum. The primary ossification center forms as osteoblasts produce bone on the surface of the calcified cartilage. The osteoblasts transform the calcified cartilage of the diaphysis into spongy bone. The cartilage model continues to grow and the bone collar thickens. Remodeling contributes to the final shape of the bone. Osteoclasts remove the bone from the center of the diaphysis to form the medullary cavity, and cells within the cavity specialize to form red bone marrow. The fourth step of endochondral ossification is the formation of the secondary ossification centers. This occurs from birth through adolescence. Osteoblasts migrate to the epiphyses and create secondary ossification centers. The events occurring at the secondary ossification centers are the same as those that were at the primary ossification center, except that the spaces in the epiphyses do not enlarge to form a medullary cavity. Bone replaces cartilage by bone until all cartilage except for the epiphyseal plate and the articular cartilage have been replaced. In mature bone, spongy and compact bone are fully developed, and the epiphyseal plate becomes the epiphyseal line. The only cartilage present is the articular cartilage at the ends of the bone. All the original perichondrium that surrounded the cartilage model has become periosteum. Bones grow in size by a process called appositional growth, where new bone is laid on the surface of bone or cartilage. Bones increase in their length when appositional growth occurs at the epiphyseal plate. In early adulthood, when growth stops, the epiphyseal plate becomes the epiphyseal line. Bones increase in their width or diameter when osteoblasts produce more bone beneath the periosteum. Simultaneously, osteoclasts reabsorb bone under the endosteum, resulting in the increase in the medullary cavity as the diameter increases. This presentation discussed intramembranous and endochondral ossification as methods for bone formation and how bones grow in length and diameter.